Hello, this video describes some examples of the types of questions that could crop up on the Paper 2 biology exam. You need to be able to recognise and name the parts of the nervous system. First of all, you have the receptor which detects a stimulus. This could be through the skin, the eyes, the nose or the mouth, which are chemical receptors. It could be via the ears, which detects changes in sound or changes in gravity, and also your temperature. You need to be able to label the sensory neuron, which transmits the electrical impulse from the receptor to the control centre. The control centre could be either the brain or the spinal cord, depending whether it's a conscious or an unconscious reaction. You need to be able to recognise and label a synapse, which is a gap between two junctions, a relay neuron, which is part of the spinal cord, and also the motor neuron, which transmits electrical impulses from the control centre to the effectors. The effectors are the parts of the body that bring about a change. This could be either the muscles, which is for movement, or it could be a gland, for example, controlling blood glucose levels. This question specifically asks you to name the synapse and the relay neuron. However, they could ask you to identify any parts of that diagram. You should also be able to describe and explain a synapse. A synapse is a gap between two neurons. The electrical message is transmitted across the gap by chemical neurotransmitters, which in turn continue to trigger an electrical impulse in the next neuron. Another skill that you are asked to show in this part of the paper is how to calculate speed. And the thing to look out for in this type of question is to check the units. This question asks you to calculate the time in milliseconds. So for any question that's a calculation, always use the every method. Use the equation that we've taught you, speed equals distance over time. From the question, we can identify the values for our variables. Information travels at 120 metres per second, so we will have to convert that. The distance is 1.6 and time is the variable that we're trying to find. The second E in the every method is for equation. So here is our standard equation. However, we would have to rearrange that, and to make t the subject of the equation, we would simply divide both sides by 120. The question asks you to put your answer in milliseconds. So you would need to multiply this by 1,000 to convert it into the correct units. The second part of the homeostasis topic focused on the endocrine system and specific hormones which maintained constant internal conditions. For example, the concentration of water, the control of the body's internal temperature and blood glucose levels. You should be able to identify and describe the hormones produced on the following diagram. The pituitary gland produces several hormones, including thyroid stimulating hormone and growth hormone. It also produces FSH and luteinizing hormone, which are involved in the maturing and the release of an egg from an ovary. These hormones are directly released into the blood, and they act as chemical messages which then target specific glands or organs to release different types of hormones. The thyroid gland controls the production of thyroxine, which controls all the body's metabolic reactions, and the adrenal glands produce adrenaline, which target the heart and the brain when the body is under duress, improving the flow of oxygen to the body. The pancreas produces insulin and glucagon. Those are the two hormones that are used to control blood glucose levels. If you do higher tier or triple, then you need to know that it's the alpha cells that produce glucagon and it's the beta cells that produce insulin. You should be prepared to describe the hormonal response to blood glucose levels, and possibly, if you're doing the higher paper, response to temperature or water levels. Whichever the case they give you, it centres around a negative feedback question. I've chosen to do a standard question which describes the two hormones that are used to control blood glucose levels in the body. So you should be able to describe what's the response if glucose levels are too high or too low. Firstly, if glucose levels are too high, then you need to name the pancreas, which releases insulin, and it targets the liver and the muscle cells. You must explain what then the target organ does. In this case, the target organ is the liver, and it converts glucose to glycogen, and therefore the blood levels drop. If glucose levels are too low, then the pancreas releases glucagon, which targets the liver. Again, I've mentioned the gland that releases the hormone and what is the target organ. Glycogen is converted back to glucose and then released into the blood. For your last mark on this question, you would then need to add in that this is an example of a negative feedback system. Generally, graph questions like this are given where you have to describe and explain the shape on the graph. In this question, it tells you that two people were being monitored to see which one of them had diabetes. It goes on to say, describe how the graph above shows that person B has diabetes. So you need to specifically refer to the lines on the graph or specific data points. Before you do a question like this, make sure you have fully interpreted the axis labels and the key. On our y-axis, we have the blood glucose concentration, which is the amount of glucose in the blood at any one time. Across the bottom, you have time, and then the key shows you the concentration of blood glucose for a person that's fasting, meaning they've had no food, and non-fasting means when they have been given food or drink. Be methodical when you're answering a question like this. What's happening at the beginning, what's happening in the middle of the graph, and what's happening at the end. In this question, we can see at the beginning of the graph, person B's blood glucose concentration level is higher than person A, and equally it's higher than the fasting range in the key. For the second mark, person B in the middle of the graph, their blood glucose level reached a very high concentration when the glucose drink was given. If you have a look, you can see person B's glucose level reached about 15.2 molecules per litre. And you can see when I've answered this question, I've specified which person I'm talking about. You've got to be specific. And then for the third mark, if we look at the end of the graph, again, person B, the blood glucose level didn't return to normal in comparison to what it says in the key. And equally, it fell slowly in the time following the glucose drink. If you're doing combined science, you can click onto the next slide, as this one is just for higher tier combined and triple science. As previously mentioned on slide 8, blood glucose levels are controlled by a negative feedback system, which brings about two possible courses of action when conditions move away from optimum. This question tests your understanding of basic negative feedback, which is, of course, when conditions move in one direction, it triggers the release of a specific hormone and therefore the concentration of that hormone. If conditions move in the other direction, it triggers a different response and that hormone, the concentration of that hormone will decrease. And it does this in order to bring the conditions back to the optimum. Before answering this question, take time to fully understand the diagram and get to the point as I have in this answer. If the concentration of substance Q becomes high, hormone A is released and therefore the concentration increases. If the concentration of substance Q becomes too low, it triggers a different response and hormone B is released. It does this in order to bring the conditions back to the optimum. I've just simply put it as hormones A and B brings the concentration of substance Q back to ideal or normal or optimum. Questions on inheritance and variation typically start with questions on the genetic material. You need to be able to recognise the size order from the largest to the smallest. You can see it goes from cell, nucleus, chromosome, DNA, gene and protein. You should also be able to recognise and explain the different parts of DNA. DNA is two polymer chains, which means it's made up of repeating monomers or nucleotides. The two strands of deoxyribonucleic acid are joined together by 4 bases, and the bases pair up complementary, C to G and A to T. It then twists to make a double helix shape, and a gene is the chemical instructions to make a protein. A triplet code is made up of 3 bases. In this question, it just asks you to name which part of an animal cell DNA is found, but it could ask you for any part of the genetic material. We have 2 types of reproduction, and you need to be able to describe and explain the 2 types of cell production and the types of cells and offspring that it produces. We have asexual reproduction, which produces 2 genetically identical cells by mitosis. These cells are diploid and have 23 pairs of chromosomes. We also have meiosis, which takes place in the gametes and produces 4 genetically different cells, and they are haploid. They only have 23 chromosomes because during fertilization, when the egg and the sperm fuse, the full number of 23 pairs is restored. This question tells you specifically that the diagram shows 2 patterns of cell division. Cell division type A is used in the gametes, so we know that at the top of the diagram here that this is meiosis, and you can see as well from the diagram that we have one cell that then divides twice to produce 4 daughter cells, which will each have 23 chromosomes and are haploid. We can then see in the second type of cell division that we have one cell that goes to 2 cells. So this is where mitosis has taken place. We have 2 genetically identical daughter cells and they are diploid. Make sure you can distinguish between the 2 types of cell production and reproduction. So to answer this question, the 2 types of cell division A and B in the diagram, type A is meiosis and type B is mitosis. When predicting inheritance, you need to be able to understand and use the following terms, which I've included here for you to look at in your own time. Please make sure you are very clear which are capital letters and which are small case letters, and also make sure when you are describing whether the combination of alleles is homozygous dominant, so it's 2 capitals, or whether it's homozygous recessive, 2 small case letters. When predicting inheritance and using Punnett squares, you must remember to ensure you show what the letters stand for that you are using to represent the condition or the feature. When you've drawn your Punnett square and you're filling it in, make sure that you make it clear which are capital letters and which are small case letters. Capital letters represent dominant alleles, small case letters represent recessive alleles. When you are filling in the grid and beforehand, write alongside it what the genotypes and the phenotypes are. For example, what the 2 capital alleles mean, that would be dominant, homozygous dominant, and therefore they would have the disorder or the condition. And then finally, make sure you also look at the question carefully to see whether it asks you to give your answer as a percentage or as a probability. This question here is about polydactyl, which is an inherited disorder, and it tells you outright that it's carried on the dominant allele and that capital D is the letter you should use to represent the condition. The question then asks you to complete the Punnett square, so make sure you fill that in fully and then identify any offspring and the genotype that would have polydactyl. So start by filling out what our letters or our alleles mean. And we can see that two capital D's here stands for homozygous dominant, which means the offspring would have the condition. We have a combination of a capital D and a small d, which means the offspring will be heterozygous, and they will also have the condition because you only need one of them. The only other combination you can have is a pair of recessive alleles, which is homozygous recessive, and those offspring with that combination will not have the disease. Be methodical when you're filling in your Punnett square. First of all, put in the genotypes of one of the parents across the top and then the genotypes of the other parent along the side. This is very similar to plotting coordinates on a graph, and we can see once we've carried out the cross, you can see that we have ended up with 50% of the offspring that carry a dominant allele and will therefore have the disorder. And we can also see that 50% are homozygous recessive, which means they will not have the disorder. The question specifies that they would like an answer in the probability, and so the probability is 1 to 1. Always show where you've got these figures from, and you can see from this it was obviously 50% to 50%, which equates to 1 to 1. I've also included some common errors to avoid, which is what we've seen when we've marked the mock papers in previous years. So first of all then, please make sure you read the question very carefully and identify the command word. So for example, is it asking you to describe, explain, compare or evaluate? Describe, say what it looks like. For example, it could be the shape, the size, the temperature. If it's talking about the line on a graph, does it increase or decrease? And any changes that you can see if any diagrams are given. If it asks you to explain, say how and why. This happens because. Use comparative language if it's a compare question. Make sure that you think about your technique before you start to answer. For example, are you going to compare point for point, or are you going to do a paragraph talking about one aspect and then a paragraph talking about the other aspect? Make sure you use comparative language. For example, this is this, whereas something whatever is the other, or things like however. Finally, if it asks you to evaluate, make sure you talk about the advantages and disadvantages, and most importantly, at the end, make sure you include what you think and why. For example, what you based your opinion on. And always use the every method when you have any kind of calculations. If you make a calculation error on your calculator, then if you've shown your workings out, you'll still get partial credit for selecting the right equation. And common errors to avoid on graph tasks. Generally, they ask you to do at least 3 things. So make sure you read the question carefully and you tick off the tasks as you do them. They generally ask you to plot the data from a table, label the axis, which you can get those labels from the labels of the top of the table, and then draw a line of best fit. Once you've plotted your points, don't include any anomalies that stand out from your line of best fit. Bypass them. Don't do a little mountain or molehill to incorporate them. It should be a straight line of best fit. Make sure you interpret the scale on both axes before you plot your data. So for example, how many little squares are represented by the scale? And make sure you plot your data and you draw your lines of best fit in pencil. If you make a mistake, you can easily rub it out. It looks better then for the person who's marking it, and they'd probably be more inclined to give you the marks if it's a clear, sharp line. And finally, some other common errors to avoid and things to check before you finish the exam paper. So when you're approaching the questions, check how many marks the question is worth. If it's for 3 marks, make sure you've included 3 specific points. So for example, if it was to describe a graph, what happens at the beginning of the graph, what happens in the middle of the graph, and what happens at the end of the graph. Be specific. Don't refer to things as it or they. That is way too vague. So for example, if you're describing the difference between a eukaryotic cell and prokaryotic cell, as you can see from the example, I've named the one that I'm talking about before I've described its feature. Always check your answers make sense after you've written them. Sometimes in haste you'll write in text speak, but please just go back and check that it is written with good English and grammar. And finally, I've included some other slides on the video that cover the osmosis required practical, eukaryotic and prokaryotic cells, and respiration, which are topics that regularly crop up on Biology Paper 1. I hope this video has been helpful, and finally I just want to say good luck in the exam.